Welcome to my second seminar. This seminar is focused on digestibility of protein and amino acids and try to understand some of the determinants that determine the digestibility and bioavailability of amino acids in feeds and feed ingredients. I prepared this presentation with the help of my PhD student, Sharari Jahambin, at the Fish Nutrition Research Lab in the Department of Animal Bioscience at the University of Guelph. There's increasing amount of information on apparent digestibility coefficient of nutrients in different ingredients due to increasing research effort. Estimates of apparent digestibility of nutrients, a variety of feed ingredients, are increasingly used by feed manufacturers who are trying to formulate their feed on a digestible protein, digestible amino acid basis, and other digestible nutrient basis. The move from formulating on a total nutrient basis, for example, total lysine basis, to a digestible nutrient basis like digestible lysine is really praiseworthy because if it's not digestible, it's of no value to the animal. However, if we are increasingly relying on estimates of apparent digestibility of nutrients or feed ingredients in the feed formulation process, we have to make sure that this information is relevant and reliable. Right? And we have to recognize or be aware of the limitation. There is also a need to better understand the basis of difference in digestibility and bioavailability of nutrients, notably amino acids, in feed ingredients. And this is something that has not been the focus of sufficient research effort, in my opinion. First of all, I like to clean up a little some of the concept. Very often when I listen about work on digestibility and digestive physiology, I find lots of mix-up. Very often people will confuse dietary habits with the ability to digest something. Let's say a grass carp can consume grass, so therefore it says, well, they can digest grass. A trout doesn't eat grass, so therefore it cannot digest grass. The dietary habits, whether it's a carnivorous or omnivorous fish species, does not necessarily implies an animal is able to digest something or not able to digest something. The carp will digest the protein and the fat in the grass and not digest the fiber. You could make a grass concentrate, a grass pellet like alfalfa meal and feed it to a trout, and it can digest the protein and the fat also of that alfalfa meal, of that grass. So therefore, dietary habits doesn't determine what the animal digests. Now, the digestive physiology and biochemistry determines what an animal is able to do or something like this. But you can actually go very fine detail and measure difference in digestibility in enzyme activities or pH or gastric emptying or something like this, which clearly have to do with the physiology of the GI tract. But these may not have an impact on the digestibility of nutrients themselves, meaning the disappearance of the nutrients from the GI tract. Digestibility is a measure of disappearance. You feed something to the animal, you measure it as being present in the feed, you collect the fecal material, and you find that this nutrient is not present or has been reduced in the fecal material. So therefore, you imply it was digestible. It was disappeared. Digestibility is not a measure of absorption. It goes hand in hand with the absorption because the digested nutrients will likely be absorbed, but it's not a measure of absorption. Measuring absorption is extremely difficult. It requires ex vivo work, cutting out a piece of the intestine and then applying all kinds of process to it, which is not very physiologically relevant or relevant for living animals. Now, often I also see that people mix up digestibility and assimilation. Assimilation is a process of utilization that implies the digestibility, absorption, and utilization of the nutrients by the animal. So assimilation or utilization is a lot more complex than digestibility and should not be mixed because in the end, assimilation or utilization involves several other processes. And then you got the position and accretion where we could be looking at equating digestibility and retention of nutrients, but the retention of nutrients is also driven by several processes. So therefore, I find it's a bit of mess out there. When we talk about digestibility, we're talking about something very specific, meaning, right, what we're measuring is a measure of disappearance. Different species will have different feeding habits, different dietary habits. That doesn't necessarily determine what they can digest. A rainbow trout can eat plant protein, a grouper can eat plant protein, and still digest these plant protein very well. So it's not because it's not part of their natural diet that they're not able to digest. Assimilation, the same way, is based on the absorption and utilization, deposition of nutrients. So therefore, it's more, right, it's more advanced than digestibility. So you have to be careful not to mix up some concept. How do we measure digestibility? Well, the very first approach that was measures is when you collect everything, right? You determine what the animal consume, or you feed a certain amount of feed, and you collect everything that is produced by the animal. Therefore, you can collect digestibility. Let's say you fed 100 grams of dry matter to the animal. You collect all the fecal material the animal produced, 25 grams. Well, therefore, 100 minus 25 over 100, digestibility of dry matter was 75%. You fed 40 grams of protein. 4 grams came out in the feces. 40 minus 4 over 40, digestibility of protein was 90%, and so on. Digestibility this way, measured by the direct method, is extremely complicated, extremely challenging under most conditions, and virtually impossible in fish. Imagine you have to collect all the fecal material produced in water, and you aerate and you flow water in the tank. So therefore, it's very difficult to collect all the fecal material and be sure that you collected all the material. The other challenge is that you can measure feed consumption over a period of time, but will the feces come out and over the same period of time? There will be a delay. How long do you collect fecal material? Well, you could stop feeding the animal after that, but then you'll end up in a fasting state in the animal. The digestion in the animal will slow down. The passage of food through the GI tract, or feed through the GI tract, will slow down. So you cannot really do this. So you have to do this, right? So there are actually a number of limitations associated with direct method, even though you could, right, if you could collect all the fecal material. Something a lot more practical is the use of the indirect method for measuring digestibility. And it relies on the use of a digestion indicator or a marker that is 100% indigestible, safe, and pass at the same rate as other nutrients. That's 3 characteristics that a marker has to have. And then, because you're using a marker, an indigestible marker, you can collect a representative sample of fecal material and actually assess the digestibility on that basis. So you incorporate your marker in the diet at a certain level, whatever marker you're using, whatever it calls for. You collect a representative sample of fecal material, and using this equation that you see, ADC equal 1 minus the concentration of nutrients in the feces over the concentration of nutrients in the diet, times the marker concentration in the diet over the marker concentration of feces, and you can measure the apparent digestibility coefficients of dry matter, protein, lipids, whatever nutrients you're interested in. Because you're using the marker, it's a marker enrichment technique. It's a substance enrichment technique, calculate. So the disappearance of the marker is 100% indigestible, becomes your reference point, and you calculate the disappearance of other nutrients by the enrichment of the marker compared to the nutrients in the feces and compared to what it was in the diet. This is a very effective technique that allows you to solve a lot of problems that was associated. You can collect a representative amount of fecal material. You don't need to collect all. You can collect over any period of time that you want, as long as it's a representative sample. So this technique has been the most widely used in aquaculture nutrition, but also in poultry and swine nutrition to measure the digestibility. Now you have to collect fecal material, and that can be quite challenging. There's several approaches to collect fecal material. There's stripping, dissection, siphoning, quite widely used in many labs. In Atlantic salmon, stripping is quite widely used in Norway and other places like this. Dissection in some species that don't handle the handling, or large species that is dissection. The problem is you have to kill the animal in order to dissect and collect the fecal material. So it can be very expensive and quite challenging. Siphoning means picking up the fecal material as produced by the animal, very labor intensive, and could also lead to breakup of feces because the feces you may not be able to siphon the material in real time. In order to deal with the limitation of the different techniques, three passive collection systems were developed and are believed to be relatively reliable. The three systems come in three families: the Tokyo University column from Japan, the Senpei system from France, and the Guelph system from Canada, from my university at University of Guelph in Canada. I show a slide here of a few systems. The Japanese have been working for several decades on digestibility, this evolution of different systems with the Nose system. And then after that, Ogino developed the Tokyo University of Fisheries system, which is basically a siphon of the water from the tank. It passed through a nice glass contraption that you fill with glass wool, glass fiber, and actually it filters out the fecal material. Very beautiful, actually really quite effective. Beside it is the Guelph system, and I'll revisit it in a second. The Senpei system, the French system, is a series of rotating screens. So you have a semi-conical bottom tank, which the fish are swimming in. The water flows from the bottom. You lift it and you bring it to the series of rotating sieves. So the standpipe, right? The water flows over. The sieves are rotating. They're in a little track here, little motor here that activate the track, and the fecal material falls, and they put a dish here, and they can collect the fecal material. You can put a freezing plate or some pile of ice there, a block of ice under the plate, and you can keep the fecal material. Very nice, but quite sophisticated, quite complicated. The Guelph system is based on semi-slope bottom tank where the fish are very comfortable, self-cleaning. Tanks connected to common drain, and there's a standpipe. The common drain has 1.5 inch diameter, about 4 or 5 centimeters, 4 centimeters diameter, about 3 centimeters diameter. And the standpipe is a 3.5 inch ABS pipe, so about a 10 centimeter, 8 or 9 centimeter diameter. There's an hydrodynamic principle. When the speed of the current, right, is high, in the volume is small, the speed of current is very fast. The feces get carried and they arrive to this area where the volumes increase, so the speed of the current drops. The flow rate is the same, but the speed of the currents drop, and actually the feces that are heavier than the water tend to settle in this, and there's an undisturbed water area. The water flows here at the standpipe, so the feces settle here, and you can collect them at the bottom of this cone-shaped thing, the sharp thing, where you can collect with a syringe or bottle or whatever contraption that you have. So you have the fecal material very effectively collected. You can collect a large proportion of the feces produced by the animal. You can look at the picture. You can see these little grain. Actually, the feces are really quite intact and not leaching out. So this system was developed by Dr. Cho and Stanley Slinger in the 1970s, and I've made this proof. All my lab at University of Guelph is equipped with these tanks, so we can carry out digestibility trial at the same time as we do growth trial. Several other labs in the world, including in China, have set up similar system or an adaptation of the Guelph system in their facility. You can see you collect the fecal material very effectively. Ever since the 1970s and early 80s, information on the digestibility of feed ingredient has been published in the literature, and you can see a lot of it comes from Dr. Cho and Dr. Slinger at the University of Guelph, carrying out some work done at Cornell University. So information on the digestibility of feed ingredients, of practical feed ingredients, have been available for about 4 or close to 5 decades. These estimates of the apparent digestibility of nutrients have been extremely useful for formulating feed on a digestible protein and digestible energy basis ever since then. So formulation of feed on a digestible protein and digestible energy basis has been possible ever since. Other research groups have contributed to measuring digestibility in different fish species, measuring the digestibility of practical feed ingredients. Work by Sean Tibbett, Joyce Millett, and Santosh Lal in Halifax in Canada measure the digestibility of several practical feed ingredients in Atlantic cod, in marine fish, and you can see they measured highly variable estimates of digestibility of protein of different practical ingredients, showing that there is value in measuring the protein digestibility of ingredients because you cannot assume that all ingredients have the same digestibility. Look at that shrimp meal, 66.7% digestible versus scrap meal, 89% versus krill meal, 96%. Uh, Basically, canola meal versus soybean meal, right? 76 for canola meal, soybean meal 92. So quite significant difference in digestibility of nutrients across species, right? Across ingredients in that species. A number of efforts elsewhere have been carried out, and the information is made available in literature. I do a fair amount of work in Asia, in Vietnam, and I like this research here by the groups of researchers in Cà province in Vietnam with the support of David Smith and Brett Lencross from Australia at that time. And you can see they measure the apparent digestibility of protein and energy of 5 different important commercial ingredients used in Vietnam. This information is extremely valuable, and especially given the fact that it was done on local Asian fish species, Pangasius catfish, right, on local feed ingredients from or imported feed ingredients available in Asia, is extremely valuable in this context. This is the kind of information that we need in for to support the industry. My lab, over the years after Dr. Cho, carried out a bunch of efforts on the digestibility of plant protein and animal protein ingredients. We've measured the digestibility of amino acids, protein and amino acids of a wide variety of plant protein ingredients, measure in general high digestibility, but sometimes you have poor digestibility, like this sunflower meal from the US. Actually was relatively quite low digestibility of nutrients. Some ingredients are extremely high digestibility. We've compiled this information in the International Aquaculture Feed Formulation Database, the IAFFD.com, right, database, which is a free database. So we compile information on nutritional composition and digestibility of feed ingredients, of common feed ingredients. We're up to 600 ingredients, if not more today, where we have on this free database. You can download this information. The NRC in 2011, this committee that was on, did a compilation of the information on apparent digestibility of protein of different feed ingredients in different species. And you can see I've highlighted in yellow values that I find questionable. You can see there's quite a few questionable values. Why would blood meal be 30% digestible in salmon and 84 or 89 or 99% in rainbow trout? What it would be 66 to 71%. Well, actually, what this table doesn't take into account is that different batch of feed ingredients have different digestibility. So it seems to suggest that the difference exists between species, but really it's a question of difference in digestibility between batches of ingredients. So several ingredients, well, you can actually have batch that have lower digestibility than other batch. So therefore, you carried out a study with a different species, you'll measure a low digestibility. It may not be a question of species, it's very often a question of ingredient quality. That table is useful, right? It paints you a picture, but you have to look at it in a more wholesome manner than simply this value for that species. You have to take 2 steps back and look at these values with a grain of salt and says, well, this paints the general picture of digestibility, but not specific picture. My review of information on digestibility of feed ingredients throughout my career and through the NRC and through this construction of the International Aquaculture Feed Formulation Database is that the digestibility of high quality ingredients, standardized ingredients, is very high. If you're talking wheat gluten, soy protein concentrate, low temperature fish meal, spray dry blood meal, very high quality poultry byproduct meal, you actually have very high digestibility, and the digestibility will be maintained across lots and batch of these ingredients. So actually, you can rely on these ingredients as table sort. You can find significant difference across species in the digestibility of certain ingredients. But you find the same kind of variation across studies with the same species, or even within studies with the same ingredients, but coming from different origin or different batch of the same ingredient. So that 10 to 20 percentage point in digestibility of protein actually may not be related to species, but really is a function of the quality of the ingredients. Variation in the quality of the ingredient. We'll get to this later on. That 10 to 20 percentage point difference in digestibility has a very important economical impact. Let's say you're formulating a feed on a digestible protein basis or on a digestible methionine basis. A 10% variation in estimates of ADC of digestible protein and methionine may result in feed costs of between $5 to $10 per ton, additional cost per ton. So actually, it has an important impact. Very often, right, in low margin business like the feed industry, at $5 to $10 a ton is actually extremely meaningful in terms of impact on your bottom line. So developing a good handle on the digestibility of the feed ingredients that aquaculture nutritionists are using is extremely important, and yet extremely complicated. When I review information on digestibility, it came across several limitations in studies and publication. I've reviewed a wide variety I've carried out a lot of research on digestibility. I've reviewed a lot of academic effort on digestibility. I've reviewed lots of industrial research and development effort on digestibility. And I've identified, notice, a series of errors. There are numerous methodological issues. First of all, there's mathematical issue. Equations used are not reliable. There's issue with feces collection, the method. There's analytical issue, chemical issues where the marker or the analysis of different component doesn't add up. And then there's statistical issue on it. Then there's nutritional issues related to the ingredient. The characterization of the ingredient origin and type can be critical, how it was produced, all this. And the other issue that I will touch upon is that you can measure certain digestibility, but the bioavailability of that nutrient may be different, may be lower than what was measured on a digestibility basis. So actually, measurements of digestibility is not always extremely reliable, and it's actually really quite challenging to make use of the information. I'll illustrate a number of concepts here. When we review information on digestibility of nutrients in the literature, we have to be very careful about making sure that this information is adding up. This is a real example from a real published study on a marine fish species from a reputable lab. They measured the digestibility of two different kinds of fish meal, and then two ran animal protein, poultry byproduct meal, and meat and bone meal, and three plant ingredient. I'll focus only on the animal proteins because in the animal protein, the energy is only contributed by protein and lipids. There's no carbohydrate. When we look at this and we predict, right, try to look at, see what the data is adding up, we can see that for example, in the case of poultry byproduct meal, the author measured close to 49% digestibility of protein. The lipids is 59% digestible, and yet the energy is close to 72% digestible. How is it possible? If all the energy is contributed by protein and fat in poultry byproduct meal, there's no fiber in there, there's no starch, there's no cellulose in there, how can the energy digestibility be higher than the protein and lipid digestibility, or the weighted average, the protein digestibility? When we compute the digestible energy content of this feed ingredient on the basis of proximate parameters, right? Digestibility of protein times the energy content of protein, the digestibility of lipids times the lipid content times the energy content of lipids, we arrive to a value of 2,500 kcal per kilogram of that feed ingredient. If we use the gross energy digestibility to calculate the digestible energy, we arrive to 3,500 kcal for that ingredient. Over 1,000 kcal difference between these two modes of calculating digestible energy. It doesn't make any sense. There's clearly a problem. And the same problem occurs for all the animal proteins. And for what we know, it could also occur for the plant proteins. You can see that mammalian meal, the meat and bone meal, the information doesn't add up also, albeit a little bit less critical, less important than for the poultry byproduct meal. Now, this is not just one of. I review the literature. I see this problem very often in publications, either paper I review or papers that are published. Is here, study out of China, where they determine the digestibility of fish meal and meat and bone meal, all this. Digestibility of protein, 92%. Digestibility of lipids, 90%. Energy digestibility, 82%, 83%. How can that be, right? This fish meal, there's no fiber there. There's no cellulose, there's no starch. So the energy digestibility should be a weighted average of protein and lipid digestibility. Same thing for meat and bone meal, 82 for protein digestibility, 70 for lipid digestibility, 70 for energy. The digestibility of energy should be higher if these values are true. So see, the data are not adding up in many of the papers. So how can we actually fully trust these values to formulate on a digestible energy basis or on a digestible protein basis, for that matter? Another example where they measured the digestibility of 13 animal feedstuff in cobia, and you can see the energy digestibility in this case is always higher than the digestibility of protein and lipids, or the weighted average of protein and lipids. The most dramatic example is 89, right, for protein, 74 for lipids, and the digestibility of 92. So these values are not dramatic, but if you're trying to be careful, right, you're looking at 10% error will have an impact of $5 to $10 per ton. Well, these data, the fact that these data don't add up is extremely problematic for formulating least cost diet. So actually, when reviewing information on the digestibility of feed ingredients from the literature, you have to develop the nose, you have to develop this know-how and ability to look at the results and determine whether they make sense or not. You cannot use them at face value. You always have to be highly critical of this information. I learned this from Professor Young Cho, who was my mentor and all this. One day I show up to him with results from analyses that I carried out, and he said, let me tell you a story. You go to the fish market and you buy 10 fish. You ask the fishmonger to cut the fish in half. You go home, you open up the bag that he gave you, and you find 11 tails and 9 head. He says, maybe you're only wrong by 10%, right? There's 1 head missing and 1 tail too much, but you got cheated. It's illogical. You got 10 fish, you cut the fish in half, you should have 10 tail and 10 head. But in the end, you end up with 11 tails and 9 head. There's a small error, but it's illogical. And this is how he taught me to look at the results. So we look at the digestibility results from this. There's actually 11 tails and 9 head in this because the results don't add up. The difference is not huge, but it is illogical, and in the end can be quite significant from a feed formulation perspective. So you have to develop this know-how to review the information critically and not believe information at face value. We use this approach when we review the information for the International Aquaculture Feed Formulation Database. Use this conservation of law of mass and energy. Nothing is lost, nothing is created, everything is transformed. So, right, was taught us Antoine Lavoisier in the 18th century France. You actually should look at results. If your protein, your mass of protein, your mass of lipids, your mass of other nutrients should sum up to the mass of this. The energy contribution of protein, the energy contribution of lipids, the energy contribution of carbohydrate should be indicative of the gross energy content of the ingredient. So everything should add up. You should actually be compiling and using data that are adding up. And you'll be surprised to see when you actually approach things this way, you'll find a lot of error in the literature that can have very significant impact. The other aspect related to digestibility measurements is mathematical error. And many years ago, I was attending a conference in Texas in 1996, and Ian Foster presented a paper saying the old equation that was developed and widely used by Cho and Slinger in the 1970s and 80s was mathematically wrong. And Ian Foster was right. actually the equation was wrong. So Foster proposed a new equation. And I started using this equation, and I was getting, it was a logical equations, but we're starting to get some errors with it. So I started digging deeper and wrote this letter to the editor of Aquaculture with my colleague Catherine Watt, where we described the process, the mathematical process. What we arrived at the end is we arrived to three equations that were all mathematically correct and logical, completely logical. So therefore, these equations, which are logical and correct, should give you the same answer, right? Well, when we applied it in reality, with real samples, with real analyses, we arrived to different estimates of digestibility with the three equations. Moreover, if we use assumed composition, we assume the composition would be this because we use a mixture of 70% of reference dyes and 30% of ingredient, mix it together and we make assumption, versus when we analyze the value, we also arrive with different estimates of digestibility. And the difference between equations and ways of doing things were not small. For the same ingredients, the same nutrients, protein digestibility, we arrived at estimates of 81.3% digestibility for the lowest value and 90.7, so close to 10% variation in the estimates of protein digestibility, simply due to simple mathematical error. Why is these three mathematical equations, which are correct, leading to different answer? Is because you're solving for one term, the digestibility of the ingredients. So actually all the errors gets piled up onto this term that you're solving for. But these different things that you're measuring has some variability associated to it. But in the mathematics, we assume that these are perfect, and they are not. They are not perfect. So actually we developed this simple simplified equation, which Segura also arrived to. By mathematically simplifying the equations, we arrived to very robust equations that gives you this equation 4, that gives you a reasonable estimate of digestibility. And it doesn't matter if you're using an expected composition or an analyzed composition, it arrives to very estimate. But you have to be careful with your marker analyses. You have to be careful with your sample analyses. Make sure they're correct, they make sense, they add up, because it will have a big impact on the estimates of digestibility. Small problem, but it can have dire consequence when you report digestibility of nutrients in this. Now, the third element is actually how you collect the fecal material. My former master's advisor, Professor Joël Delanoux, in Laval University in Quebec City, Canada, and my colleague and friend Grant Vandenberg, who did a PhD with Joël Delanoux and now is a professor at Laval University, carried out a trial when they actually look at 3 fecal collection methods and 3 different digestibility indicators or markers to look at the digestibility of one single diet, a good quality reference diet that they formulated to this. That's a really interesting studies. So they had installed the Saint-Pé system. Joël Delanoux was one of the creators of the Saint-Pé system. They installed a Guelph style column. Grant came from Guelph and he basically brought this knowledge with him. And they also used stripping, which is commonly used in Europe and in Norway to measure digestibility of feed in salmon and fish species. And they used 3 markers: chromic oxide, acid insoluble ash, and titanium oxide. Interestingly, the 3 approach to collecting fecal material resulted in different estimates of digestibility. The Guelph system resulted in a higher value, suggesting perhaps too much leaching. The stripping method results significantly lower value, suggesting maybe the feces were not completely, the feed material was not completely digested. And the Saint-Pé system result in somewhat middle value, something. So that would actually perhaps less leaching and perhaps deeper. Interestingly, the 3 markers led to different estimates of digestibility. In many cases, even using the stripping technique, you can see the titanium oxide results in higher estimate of digestibility, and not a little bit, right? Over 16 percentage point, in this case, probably 30 or 40% difference in digestibility between titanium oxide and chromic oxide for dry matter digestibility within one system. So actually, not just the feces collection system can have impact, but also the marker that you're using. We assume the marker is 100% indigestible, safe, and pass at the same rate as other nutrients. And it may not be completely true. There may also be issue with analyzing these markers. When you have high ash content, you ash your sample, silica will fuse and actually will bind some of the markers and make them very difficult to solubilize and will affect the recovery of your marker. So you have to be there. The take-home message is digestibility estimates depend on the system that you're using to collect the feces and the marker you're using, and depends on the nutrients you're focusing on. So when people report digestibility value with 1 decimal or 2 decimals, I never believe this information because the information is so variable. The accuracy of the measurement is really quite poor. You cannot go to second decimal, no way. Even first decimal, I don't believe it. You should round up your value as much as possible. Now, I show you all the bad side of things, right? Say, well, the different technique doesn't leads in different estimates of digestibility, the different marker leads different estimates of different mathematics leads different estimates of digestibility, different batch of the ingredients. So why measure digestibility if it's so complicated, so fraught with error? Well, it's a bit like this lady, right, waiting for the perfect man on a bench. You actually cannot wait all your life for perfect digestibility technique or perfect man. You have to basically make do, right? You have to accept reality that no technique is perfect, but can be useful. At the University of Guelph, we've been using the Guelph system consistently over 5 decades with the same approach, and actually it led to very interesting information on the digestibility of nutrients. We're able to measure difference in digestibility over time in a number of quality ingredients, notably the land animal protein, poultry byproduct meal, fatty meal, blood meal, meat, and bone meal. We've measured very significant improvement in their digestibility over time. Now, I mentioned at the beginning of this seminar that digestibility and bioavailability is often confused together, and they should not be confused together. Digestibility is a measure of disappearance. It was in the feed. You collect the fecal material. It's not in the fecal material, so therefore it disappeared. You say it's digestible. You assume it was absorbed, and you assume it was useful and usable by the animal. Bioavailability is a measure of usability of the nutrients. So it's one step further than digestibility, and it's really quite different. Now, I have a colleague at University of Guelph work with pigs, and they actually installed these cannula, T cannula in the ileum of pig to collect ileal material to measure the digestibility in these animals. They actually measure very significant apparent digestibility across feed ingredient. They measure higher digestibility in soybean meal, lower in canola meal, lower in DGGS, and somewhat intermediate in wheat shortness for these 4 essential amino acids. So they've characterized the digestibility of amino acids in these relatively standard commodities available in Canada for feeding swine and around the world. When they formulated feed on a standardized ileal digestibility basis, so they standardized all the nutrients on that basis, make sure that the lysine requirements was met and the threonine requirement was met in both cases. Actually, they found that the formulating the feed on the same standardized ileal digestibility, amino acid digestibility or digestible amino acid content could result in different performance. In this case, casein, animal protein, was doing a lot better than wheat short, even though the diet were formulated to the same level of digestible amino acid, indicating that actually apparent digestibility may not be a good predictor of bioavailability of amino acids. You have to be careful about that. You can formulate on the same digestible amino acid basis with the best information, but that digestibility may not translate into a good measure of bioavailability. So you have to be careful about that. We were aware of this when we work with fatty meal. We actually measure very significant improvement in the digestibility of fatty meals over the years using the Guelph system. I measured in 1999, published in 1999, digestibility of 81 to 87% digestibility of fatty meal in rainbow trout compared to 58% in the 1970s and 80s by Professor Young Cho and another author. So you can see estimates of digestibility is really quite highly variable across this ingredient. We actually look at the digestibility of feta meal and found that the digestibility ranged between 60 and 90%. But besides this showed difference in digestibility, even in highly digestible one, we did find variability in the nutritive value of different batches and suppliers of feta meal. In some case, and show you results, we've used up to 25% feta meal in a diet of rainbow trout, and performance was excellent. I've seen similar trials with salmon with 12 to 18% feta meal, and actually salmon performing extremely well. In other trials, these level of feta meal resulted in lower growth performance, all this, and we never quite knew it. We found very limited correlation between digestibility and actually the nutritive value of feta meal, and we never quite understood why. In some case, some feta meal, you increase the level of feta meal in a diet of a carp, for example, a poor quality feta meal, and it's like increasing sand. Actually, 0.369% feta meal increase the FCR of common carp as if this feta meal was completely indigestible. In other case, we got excellent results with feeding feta meal, like I mentioned, 25%, 18% feta meal with absolutely no problem. So actually, we have a good handle on the digestibility of feta meal, but we don't have a very good handle on the bioavailability of amino acid. And that led me to think about what determines whether something is digestible and whether something is bioavailable. People assume it's often the species and the rental condition, and actually what we found out has little to do with this. Everything has to do with how the ingredient is processed. It's related to the chemistry of ingredients, and hopefully I'll show you about this. Now, in terms of difference between species, I've reared numerous fish species in my lab, fed the same diet, and you can find some small difference in digestibility. They can be significant, but the order of difference is 2 or 3% difference, sometimes 1% difference in digestibility. So I don't want to say difference between species do not exist, but overall the differences are very small. If a protein is of a high quality, it is highly digestible to a wide variety of species. If a protein is damaged, it's poorly digestible to many species. A shrimp, a carp, right, a sea bass, or a rainbow trout, a salmon, it will be poorly digestible for those species. So species has a very limited amount of this. Right? You can actually use digestibility in one species to predict the digestibility in other species. That's what's done in Norway, for example, where they use mink to predict the digestibility of different fish meal for chicken and other animals and salmon. You can see they related the mink digestibility with the chicken digestibility in chicken, and they find really quite high degree of correlation between two measurements. The values are a bit different, but they actually rank in the same way. So actually, difference between species are not that big. It's more a question of the ingredient quality themselves, the processing of these ingredients. I got insight on this working on rendered animal proteins many years ago, measured the digestibility of dozens of batches of these ingredients. And the most fascinating was blood meal. You buy blood meal, you get different drying techniques are used to dry that blood meal. You buy spray dried blood meal, it's extremely digestible, completely digestible, close to 99% digestible for many spray dried blood protein products. You buy a ring dried product, quite highly digestible, revolve around 85 to 90% digestibility. Then other kind of drying technique like steam tube and rotoplate dry is more highly variable, and you'll find in the upper 80s or around 80% digestibility of both protein and energy. It really depends actually how this equipment is operated. I met engineer at rendering plant, they're telling me they have rotoplate dryers and they can use it a certain way where they can end up with a very highly digestible product. So it's very much in the operation of this equipment, not the name of the equipment themselves. The same thing can be seen with plant protein, in this case, rapeseed meal from data from France, from 29 samples from 6 crushing plants, where they measured lysine digestibility. There was no correlation between crude protein content and lysine digestibility. It all had to do with the plant. You see, they group by plant. Some plant had maintained high digestibility of lysine. Some plant maintained low digestibility, lower digestibility of lysine, regardless of the protein content. So processing condition at specific plant play an important role in the digestibility of nutrients. So it's really a question of processing, not a question of species, not even a question of feed ingredients themselves. The main processing occurring is a thermal treatment. Why are we using thermal treatment, thermal processing with feed ingredient? Well, if you're producing rendered animal proteins, you have to prevent spoilage. You have to reduce the volume. You're dealing with wet material, guts and piece of muscles and things like that. You have to drive off the water. You have to separate the fat and the proteins and the minerals. So you have to use heat. You have to sterilize this product, right? By drying, by reducing the volume, by dehydrating it. You prevent spoilage, you reduce volume, all this. In the case of some plant protein ingredients, you have to reduce anti-nutritional factor. For example, soy. You cannot feed raw soy, you'll have a problem. You heat treat the soy, you steam the soy flakes in order to destroy the anti-nutritional factor or reduce them to a level manageable enough. So heat processing is central in the processing of feed ingredient. It's all a question of balance though. You want to process your ingredients sufficiently so it's not under processed. If you under process, there will be high level of moisture, high risk for high level of anti-nutritional factor, high risk for microbial spoilage, problem of handling and storage, caking and bridging and all these different things. However, if you overdo it, you have risk of over processing, creating heat damage, creating heat change inside the protein and the amino acids, destroying the amino acids or rendering them unavailable, and lowering the nutritional value of your feed ingredient. So it's very much a matter of balancing the heat treatment, processing, thermal processing of feed ingredient. You want to find the sweet spot that optimizes, right, that allows proper processing to deal with the limitations, but not overdo it, not to cause any damage. You can create heat damage in protein. This example from soybean meal. You can actually significantly decrease the digestibility of lysine with excess heat. You put soybean meal in autoclave for 15 to 30 minutes and the digestibility of lysine goes down linearly over this treatment. This lysine becomes unavailable. It destroys and not available for the animal to grow. So you can see with the heated soybean meal, you get less weight gain, you get less good feed efficiency, higher feed conversion ratio with this. So we can actually destroy amino acids using this process with heat damage. So balancing the heat treatment is key, is central. Going back to the data reviewed by NRC, you can see for blood meal, reported for salmon at 30%, 66-71 in shrimp, and 84 to 99% this. Clearly, we're dealing with different batch of the same ingredient. Blood meal can be produced in different ways, so it's not unheard of having a low quality blood meal because it was not prepared. It was not dried properly. It was dried too harshly. If you have a high quality product, the digestibility is very high, as illustrated in the work with rainbow trout, silver perch, steelhead sea bream, rockfish, where the digestibility is over 84, 85, 90% in these species because good quality blood meal was used, not overheated thing. The same thing can happen for canola, the same thing can happen for feather meal, fish meal, etc. So actually, heat damage can occur for feed ingredient. Now the question is, how does chemical processing affect protein and amino acid? We say, okay, heat damage by heat, but what kind of chemical change is associated with thermal processing and heat damage? What is heat damage chemically speaking? And then what are some of the consequences of the exchange on the nutritional quality of these protein sources? It can be damaged, but will it have a significant impact? Sherry or John, my PhD students, uh, helped me review the literature on heat damage to amino acid. And she identified 5 kinds of damage associated with heat, 5 kinds of heat damage. First of all is protein oxidation, free radical formation, protox for short, pyrolysis of amino acids and carbohydrate, racemization of amino acid reducing carbohydrate reaction called Maillard reaction, and then protein cross-linkage, protein to protein cross-linkage involving amino acid cross-linkage amino acids, or linkage of amino acids between linkage between cysteine and cysteine to create cysteine, right? So the dimer of cysteine to cysteine. We'll revisit this. Now, protein oxidation is driven by free radical, creates reactive oxygen species, reactive nitrogen species, and these species will directly damage amino acids, fatty acids, will create oxidized sugars, and all these things. These changes are irreversible. They're a chain reaction that destroy things, and lysine, arginine, methionine, and threonine actually are sensitive to these kind of things and will create carbonyl residue. You can actually lose sulfhydryl group to this, and you can also create cross-linking of protein using this Protoc reaction. We'll revisit this in a moment. Pyrolysis, what you see when you do barbecue, right? You cook under high heat, right? Under a flame or something like this. It's a high temperature process. It actually results in a thermal decomposition of amino acids. So the amino acids get directly destroyed, right? They basically create cyclic product, pyrrole, benzene, benzonitrile, quinoline, inidine, and pyrene, and like. So the amino acid get destroyed by the intense heat from the processing. Common when you're barbecuing meat, but is it common when you're processing feed ingredient? Unless you go to the extreme, right? Probably fairly unlikely, unless you have a drum dryer that's powered with flame or something like this. In the case of system for drying fish meal and blood meal in the past. So these pyrolytic now carbohydrate can be involved in this process, right? In this pyrolysis, and not just amino acids, but carbohydrate can be involved in the process, and it creates these PAC and PAH in the process also. Now, amino acid reducing carbohydrate reaction, also called Maillard reactions, are really quite important and quite actually for a lot of people who are focusing on them. They're the same reaction when you're baking a product, you're making a cake or a bread or something like this, the crust becomes nice and brown when you're cooking things, right? The reducing sugars, glucose, fructose, are reacting with amino acid. It's a complex chemistry. It occurs at a certain temperature. So for example, lysine is very sensitive to this. Will react with a reducing sugar like glucose or fructose, create a Schiff base, and this reaction is reversible. Actually, you can free up the lysine from that Schiff base. However, when it proceeds to one step further to the amidoride product, it becomes irreversible. And then there's a series step, and you end up with the formation of carboxymethyl lysine and a whole variety of this. So you actually have advanced cyclization product or Maillard reaction product of different kinds, depending on your starting point, your starting material, whether lysine, another amino acid, your starting reducing sugar, and how far it progressed in the reaction of this. So it's really quite complicated. Now it's involved in the formation of flavor in food. So it's an important reaction from a food chemistry perspective. But from an animal nutrition perspective, it actually a bad series of reactions. You can see the evidence of Maillard reaction product by looking at soybean, for example, that you heat treat. The color will change. They're called also browning reaction. So these compounds have brown color, a dark color, and you can see your product change color as you heat treat it. Indication, right? The same way you bake a bread or something, you start with something that is really white and it turns out into golden, right? Golden color. It means actually that there's some Maillard reaction product being created. So that's an important type of reaction. Now, how important Maillard reactions are in affecting the nutritive value of ingredient. Some amino acids are more sensitive, lysine, and a significant amount of lysine can be lost to Maillard reaction. Now, the other type of reaction is the racemization of amino acid. Amino acid can be found in nature under the L form. Sugars are found under the D form. Amino acids naturally present in L form. However, D forms are present, synthesized chemically, and they're present in nature to this kind of high temperature processing. So alanine can be transformed into D-alanine through this process of heat damage. D-alanine may be bioavailable or may be less bioavailable. There's not a whole lot of study. We know that D-methionine is as available as L-methionine, so probably the D-alanine is still bioavailable, but we're not exactly sure. You have to have an D-isomerase for that specific amino acid to be present in order for the animal to utilize it. The importance of racemization of amino acids on the impact of nutritional value is unqualified, not very well studied so far. But it is something that occurs. If you actually treat a test protein under condition, you can actually create significant amount of isomers of these amino acids, and they will be favorized by high pH reaction and higher temperature, longer duration of the process. So there is significant formation of the isomers of amino acids during heat damage, especially under specific condition like this. The other, there are very few studies on the impact of the amino acids on digestibility, only kind of correlation. This study here look at the processing of fish meal, standard fish meal processed according to different conditions or different grades of fish meal, standard fish meal, low temperature processed fish meal, and then different feeds made. They measured the digestibility in mg, and they found a relatively good correlation between the level of D-aspartic acid and the digestibility of this, but they also found a linear association between reactive sulfhydride groups and digestibility. So we're not quite sure what determines whether the isomers of amino acids affect digestibility, but it seems to be an indicator of potential problem. Now, if we look deeper, the chemistry is extremely complex. They actually can be, the fifth reaction is protein cross-linkage. And protein cross-linkage is done through amino acid cross-linking. Protein are chains of amino acid, and the amino acid within the protein can create these links. They can link with one another. A very important cross-link is cysteine. Cysteine is the dimer of cysteine plus cysteine. So cysteine, S-Y-S-T-I-N-E, is an isomer of cysteine, S-Y-S-T-E-I-N-E. Right? It's a dimer of two cysteine molecule. So that's a cross-link amino acid. Why is they're created? It's called disulfide bridge. It actually provides protein with very strong stability. Now, interestingly, within protein, other amino acid cross-link, or at least different metabolite of amino acid can cross-link. Cysteine can, under high heat condition, a specific condition, decompose into pentocysteine residue and dehydroalanine. Serine, common non-essential amino acid, can be dehydrated and transformed into dehydroalanine. This dehydroalanine is a molecular glue. It loves to bind to other molecules, but it doesn't bind by itself. This dehydroalanine is created within the protein. So it actually attracts other parts of other protein and create these cross-links. Dehydroalanine will react with ornithine. It will react with histidine. It will react with lysine. It can also react with cysteine to create lenthinine, with lysine to create lysinoalanine, with ornithine to create ornithoalanine. Not something you find in protein, but it can react to histidine, creating histidoalanine. These linkage are actually created within the protein by these residues. So actually, cysteine and serine can give rise to dehydroalanine. Dehydroalanine has this affinity for cysteine, lysine, ornithine, and histidine, creating these cross-linked amino acids within protein if these are located within the protein. So actually, lanthionine, lysinoalanine, and histidoalanine can be created within protein. You can also have binding with ammonia and create beta aminoalanine. You can also have a decomposition of cysteine into cystic acid, which also can lead some other problems. You can study experimentally the formation of cross-linked amino acid in vitro under different conditions. As you increase temperature, you can see very significant rise of lysinoalanine, right? And in this sample, right, with increasing heat applied to the sample, and in parallel, a decrease in the lysine content of that sample. But not just lysine, you can see arginine is disappearing, serine is disappearing, and threonine is also disappearing at the same time. So in this study, they focus on lysinoalanine, which is a byproduct of reaction of dehydroalanine and lysine. But you can see there's indication that other molecules get also consumed, right, transformed in the process. It's simply that we're not looking for those individual marker of this. As a consequence, actually, when you look, this study was carried out a long time ago on feather meal. They use different pressure, different pH, where they process raw feathers into feather meal. As they increase pressure or temperature, they also increase the formation of lanthionine, that cross-linked amino acid. The higher the pressure, the higher the pH, the more formation of lanthionine, which is a reaction of dehydroalanine with cysteine. All this, an indication of cross-linked amino acid. This does not occur by itself. It occurs within the protein. It creates cross-linkage within the protein, and then that's important to take into account. So how does it work? When native, undamaged protein, not damaged by heat, is a long chain like this. It can be in nature, but it's basically when you idolize it using a protease, a series of proteases in the gut, you end up with a series of easily hydrolyzable peptide, dipeptide, tripeptide, individual amino acids. Pretty much the protein get all chopped up into small pieces that can be digested, handled by the enterocytes, right? Dipeptide, tripeptide, individual amino acid. When you create cross-link within the protein, you create knots within that protein, physical knot. I like to think about that knots in your shoelace. Take your shoelace, makes a series of knots, complicated knots into them, and then try to cut your shoelace into small pieces, and it will be very difficult. There's going to be pieces you're not going to be able to chop very small because of these knots. So when you have heat damage, you have the formation of cross links to the protein, and you end up with a mixture of easily hydrolyzable peptides, dipeptides and tripeptides and stuff like that. But then you end up with these polypeptides, which can be relatively short and soluble, but they're capped off by these cross-linked amino acids, and they can actually be refractory to digestion. They can actually be difficult to digest by the animal. This can be disulfide bonds, cysteine to cysteine linkage, or it could be linkage between dehydroalanine and with lysine, dehydroalanine with histidine, and the like. The formation of cross-linked amino acids can have significant impact. For example, the formation of lysinoalanine will reduce the concentration of lysine. It will consume lysine. So it will reduce the nutritive value of feed ingredients by reducing the lysine content. It can decrease protein digestibility because the formation of these, polypeptides capped off with cross-linked amino acids, they will be resistant. The protease will arrive there and it will stop doing its job. It will stop because of steric hindrance by the other amino acid that's linked to the amino acids and the protease wants to hydrolyze. So it blocks proteolytic site. And in some case, it can actually be toxic. Lysoadenine has metal binding properties, and there's some evidence that leads to pathological change in the kidney of rat and potential humans, and who knows, in fish and crustacean also, because of their characteristic. So the formation of crossing amino acid destroys amino acid, reduce the digestibility of the protein, and can actually have toxic consequences. So it's something. You can actually prevent the formation of cross-link by adding material that will react with the amino acids not within the protein. So you had stuff, the cross-link amino acid will be created, but will not be created within the protein. So therefore the amino acid will get damaged, but it will actually not create these not within the protein, and it can be more easily adding things like cysteine and acetylcysteine or reduced glutathione to a mixture of soy protein and you process actually significantly reduce the formation of lysoadenine within the sample. So these are strategies that could be used for improving the nutritional value of protein, right, or maintain the nutritional value of protein when we heat process them. Okay. Now, the other important protein cross-linkage I mentioned is the creation of cysteine disulfide bond, right? The cysteine to cysteine molecule. So the two cysteine binds together, have very strong affinity together. Their sulfidary group will align and actually bind and create a disulfide bond. This disulfide bond is a very stable bond. It's used in the formation of keratin, what is present in hair and feathers and nail. Why is keratin so rough, so hard? It's because of the presence of these disulfide bond that creates stiffness, great stiffness within this molecule, and prevent these molecules from being easily digested. Actually, there's some evidence by Osved 1984 that if you heat treat a protein, you can actually increase the level of disulfide bond within this and reduce the level of sulfide group within your sample. So there's some people that think that heat processing can increase the level of disulfide bonds in the sample and thereby make the protein less susceptible for this. Now, what impact does these native or residual or newly formed disulfide bond on protein digestibility amino acids? Well, actually, one of my students, Guillaume Futsi, works in the rendering industry today, carried out the whole thesis looking at this issue. What impact these disulfide bond had on the digestibility of feather meal, a commercial feather meal, to rainbow trout as its model species, but he also worked on broiler chicken. Feathers, right, beaks, claws, and all this are important raw material for the rendering industry. A lot of it is transformed into feather meal. Feathers, but also hog hair, can be transformed into feather meals and mixtures like this. They're an important resource. They're very high in protein. So therefore, they're great interest in using these raw material to produce feedstuff. The problem is keratin-rich product like this are poorly digestible under their native form. If you feed raw feather or raw hog hair to fish or to pigs or to chickens, these materials made of 90% keratin, you actually measure digestibility of 0. I got a good friend in grad school who was doing this with pigs, ground up raw feathers, fed it to pigs, and it came out the other end of the animal, 0% digestibility, absolute 0 digestibility of the raw feather. Now you take the feather and you steam hydrolyze it, like it's done, right, with steam hydrolyzed feather meal produced by the rendering industry, and the digestibility becomes really quite good. I mentioned between 60 to 90% digestibility, right? Basically, over 70% digestible routinely is achieved to simple steam hydrolysis of this. Why? It's because the steam hydrolysis break the disulfide bond. Now, you purchase feather meal, and there's still some amount of residual disulfide bond present in this. The steam hydrolysis does not completely destroy all the disulfide bonds, and some disulfide bond may actually be recreated during the drying, during the processing and drying. So we were interested in understanding what impact the disulfide bond that was present in the feather meal could have, and if we could do something about it. So the first step was to try to destroy these disulfide bonds that were remaining. So Guillaume and other colleagues worked on a novel enzymatic treatment to increase the digestibility of feather meal, commercial steam hydrolyzed feather meal. And how it works is basically started with feather meal, and he treated with sodium sulfite, Na2SO3, that actually reacts with the disulfide bond and destabilize the disulfide bond, open up the disulfide bond. And then after that, so the cysteine, right, plus the sulfite, creates a Bunty salt, and then cysteine. So actually one of the cysteine within the cysteine residue within the cysteine dimer becomes unstable. And then Guillaume used a protease, a commercial protease, to hit the feather meal, right, the keratin, when this disulfide bond was destabilized, thereby idolizing it, the protein, while it was susceptible to degradation by protease. You use a protocol. We use different level of protease, different level of sodium sulfite, and different water to feather meal ratio in a factorial design. And that was very interesting. It's actually the enzyme was essential. If we didn't use any protease, nothing happened. But the combination of protein and sodium sulfite greatly increased the degree of hydrolysis. So we measure degree of hydrolysis, right, by solubility of peptides, and actually the digestibility, the solubility, the degradation of protein greatly increased with increasing level of sodium sulfite. A small amount of enzymes was required, but adding more enzyme didn't do anything. It was really the sodium sulfite that was the trick. And the amount of sodium sulfite that was required was equivalent, the molar equivalent, to the amount of cysteine that we wanted to free up. So actually we had to use up to basically 2.5 to 3% sodium sulfite in order to idolize, to destabilize the 2 or 3% cysteine in the disulfide bonds in the feed ingredient. So that's very interesting. That led to a creation of a very nicely idolized product, which Guillaume used and fed to rainbow trout and also to poultry, to broiler chicken also. And look at this. And he published a series of papers on different aspects of this. So he scaled up the process, produced kilogram quantities of these feather meals, started with a feather meal that was a mixture of hog hair and feathers, started with feather meal as pure feather, commercial, from 2 different commercial plants, produced a highly hydrolyzed product. Pre-treated feather meal 1 and pre-treated feather meal 2 became extremely flowing. Actually, the smell became really quite nice, really quite pleasant to things. The product became extremely fine through this process. He measured the digestibility of nutrients and amino acids in these feather meal, either intact, original, or pre-treated, right? Original and pre-treated, and found that the digestibility of amino acid increased extremely significantly across the treatment, the pre-treatment. Arginine increased from 86 to 95. Histidine went from 50 to 100. Isoleucine from 86 to 94. You see the idea, right? In both feather meal, the digestibility of protein, the digestibility of amino acid increased very significantly across with the pre-treatment. But this is digestibility is a measure of disappearance. It's not a measure of utilization. So therefore, Guillaume carried out a bioavailability trial where he looked at the response of the growth of animal in response to increasing level of nutrient provided by a standard source. In this case, we use arginine, we use L-arginine, and compare it to, right, the ingredients, right, graded level of ingredient. This is the response of the animal, right, to L-arginine, to the increasing this. You can see it's a little bit difficult to see, but this is the improvement due to the pre-treatment of feather meal 1. So the arginine retention went from 55% up to 65% or from 62% to 70% with the heat, with the processing of it, making the arginine more bioavailable by destroying the disulfide. The arginine is not present, is not part of the disulfide bond. It's an amino acid right within the protein, and the digestibility of arginine was improved by hydrolyzing the disulfide bond, indicating that these disulfide bond that was present, the residual disulfide bond that was present in commercial fatty meal, was impeding the digestibility of other amino acids and making that protein source more bioavailable. We fed it to rainbow trout, obtained terrific growth performance, excellent palatability of the diet. Now, exceeding at high level, the performance of the animal dropped on this because the balance of amino acids became less good. But in this case, there was about 10 to 15% fed meal in the diet, and the performance was absolutely excellent out there. Now, why would there be, now it was interesting to see that there's difference in digestibility between the fed meal 1 and fed meal 2. The fed meal 1 was more highly digestible than fed meal 2, but when we compare performance, fed meal 1 was performing a lot less well than fed meal 2. So what was happening here? Why would a product be more digestible, perform less well than a product that is less digestible? Well, amino acid digestibility is not a measure of utilization, it's a measure of disappearance. And what seems to be occurring is that fed meal 1 contained more cross-linked amino acids than fed meal 2. So actually cross-linked amino acids may be a good indicators of protein bioavailability of amino acid more than digestibility of protein. So the difference in quality of fed meal seems to be related to the presence of cross-linked amino acid. Now, Guillaume used very advanced technique to look at the impact of disulfide bond, the type of disulfide bond, to quantify them. He developed an Raman spectrometry technique to measure the disulfide bond, identify an area, identify the disulfide bond using this very advanced laser-based technique, and actually found a way to quantify the amount of disulfide bond and the conformation of disulfide bond using advanced analyses and advanced mathematics. What he found is that disulfide bonds are actually found in two different conformations. Trans goes trans, goes trans goes, and goes goes goes. This basically the orientation of the molecule. Even that you have two molecules, they can be oriented and give three kinds of orientation. Actually, he found that the processing with this technique, this disulfide, the breakage through the sodium sulfate and protease, actually reduced the amount of certain types of bonds and increased it actually increased the amount of more susceptible, easier to hydrolyze disulfide bond. Becomes really quite complicated chemistry, but you can read these papers. It's really quite interesting, and was able to look at the quality of fed meal on the basis of the characterization of the different bands within this. So the same way cross-linked amino acids are creating bridges, right, within the protein, disulfide bond can create these bridges, create these, right, these molecules that are more difficult to hydrolyze because there's knots in them, create these polypeptides that actually can be quite resistant to digestion, but can be quite soluble. So actually when you measure them using digestibility technique like the Guelph system, you could actually be measuring them as digestible because they may leach out in the water because they're quite soluble. So you'll measure them as being digestible because this disappeared, but they were not usable by the animal. So that's one of the reasons why you have to be careful about digestibility measurements because of this potential disappearance of some molecule that you measure as digestible and may not be bioavailable. But even more than this, when we look at the work of Latcha from 2000 and look at pepsin digestibility, you can see that pepsin digestibility increased the same way that lanthionine increased in digestibility. So you actually can be measuring something as highly digestible using pepsin digestibility, but could contain quite a bit of cross-link amino acid. Remember, when you're measuring pepsin digestibility, you're not hydrolyzing things to individual amino acid. You're hydrolyzing things to polypeptide that are small enough to go through a filter paper, but not hydrolyzing them to small peptides or individual amino acid. So actually, the polypeptides that are resistant to digestion may be measured as digestible according to pepsin digestibility, but may not be digestible or fully digestible and bioavailable to the amino acid. So it's an artifact of this. So in general, I do not trust all that much pepsin digestibility to measure, to assess the quality of protein because of this type of artifact. So remember, something could be difficult to digest, could be highly water soluble. You could be measuring it as highly digestible using the Guelph system or pepsin digestibility assay, and yet this stuff may not be bioavailable to the animal. So actually, it's a really quite important. Now, Sherry Jahan, and my PhD students continue on that path and look at the different chemical change that occur during thermal processing of protein ingredient. So she used a model protein, an animal protein, chicken breast meat, and she selected this because there's low level of blood. There's a little bit of blood, but not that much in breast meat, right? There's low level of fat. So she freeze dry, very easy to freeze dry and grind into a very fine powder that you can use as the model protein. And it's just not like casein or albumin. It becomes really quite practical protein source that can be relevant for the rendering industry and the meat processing industry. She actually stimulated the formation of different types of chemical reaction. She first used no supplements, no agent in it, and she cooked the chicken meat meal in chicken meat powder at increasing temperature in an autoclave. She wanted to stimulate protein oxidation, so she had ferrous sulfate, iron +2, into the mixture that would promote free radical formation to the donation of electron by ferrous +2. And she also stimulated cross-linking of amino acid by adding L-serine, which will decompose into dehydroalanine, and treated the chicken meat powder the same way at increasing temperature. All this. This is the end result. She had moisture, she had her agent, she autoclaved the stuff, and you can see the significant transformation, the change in color of the product when increasing this. But she didn't limit herself to color. She looked at very specific chemical indicators. She looked at DL-lysinoalanine in there. And you can see that in the presence of no agent, right, you had very little formation of dehydroalanine, but in the presence of reducing agent like ferrous sulfate or L-serine, free L-serine, you had very significant formation of lysinoalanine, especially at temperature exceeding 110. So 120, 130 degrees Celsius, you had very high formation of lysinoalanine. She looked at the formation of lysinoalanine, both DL and LL, lanthionine, and beta-aminoalanine, BAL. And actually, you can see the formation. The BAL didn't result much formation, but you can see lanthionine shoot up. The formation shoot up above 120 degrees Celsius. Lysinoalanine started being produced above 110 degrees Celsius. Very significant, especially in the presence of this cross-linking stimulating compound, L-serine, that gets transformed into dehydroalanine. So remember, the serine gets transformed into dehydroalanine, and the dehydroalanine reacts with the lysine to create lysinoalanine. We've seen this. The dehydroalanine reacts with ammonia. We didn't see this because there's not much ammonia in our product. It will react with cysteine to create, right, lanthionine, or the pentocystic residue is going to react with cysteine, create lanthionine. We saw this increase. Now, we could not measure histidoalanine because the standard, basically the elution of the histidoalanine through HPLC was very similar to other molecules. It was obscured in the analysis, so we could not actually detect the formation of acidoalanine, even though we had the standard. So actually, this process, right, of the formation of cross-linked amino acid actually may be responsible for difference, right, in digestibility and bioavailability of feather meals of different origin. You can see that feather meal is 86% digestible. This feather meal is 85% digestible, right? Same digestibility in this case, diet, in the diet, different performance, meaning that some amino acids may be digestible but not bioavailable. There's a difference in the performance. So actually, we look at feather meal 1 and feather meal 2 here. There's 125% more lanthionine in feather meal 1, 260% more DL-lysinoalanine, and 280% more beta-aminoalanine in this ingredients compared to feather meal 2. So we think that cross-linked amino acids may be an interesting indicator of heat damage, or potential past heat damage, and indicator of bioavailability of amino acids in this. So in summary, for this lecture, information on apparent digestibility and nutrients available for an increasing number of feed ingredients. This information needs to be reviewed carefully because not always reliable and relevant. You have to be careful. Something can be measured as digestible, but may not be bioavailable and usable by the metabolism and the animal to grow. You have to be careful about this. A measure of digestibility is a measure of disappearance, not a measure of usability. It's useful, but not perfect. Thermal processing that is used in the production of most ingredients, many feed ingredients, has a major impact on protein digestibility and especially bioavailability of amino acids. We have to be very careful about this. And we need technique in order to assess those. And we think that the measurements of cross-linked amino acids or the measure of disulfide bonds using, for example, Raman spectrophotometry can be extremely useful. Amino acid cross-linkage results in formation of lysinoalanine, disulfide bond, lanthionine, which affect the digestibility of amino acids, the bioavailability of amino acid. There's definitely need to be better quality assurance and quality tests to assess the nutrient value of protein more than a pepsin digestibility, another traditional technique. Now, there's something that can be done about it. When processing, when producing land animal protein, rendered animal protein, some fish meal, even soybean meal, some additive could be used in order to prevent the formation of cross-linked amino acids within the protein and therefore maintain the digestibility and bioavailability of these of overall amino acids present in the feed ingredient. There's different, after the fact, there's also solution we've demonstrated with feather meal to a simple treatment with sodium sulfite and a commercial protease. We could actually tremendously improve the digestibility and bioavailability of nutrients in these feather meals, commercial feather meals, with this simple treatment. So there's hope in order that to be able to deal with it. I wish to thank the North American Renderers Association, especially Dept, for his support, the Fats and Protein Research Foundation. A lot of the work was supported by Evonik, NSERC, National Science and Engineering Research Council of Canada, Natural Science Engineering Research Council of Canada, the Ministry of Agriculture, Guelph Nutrition, Sanimax, USEC, Guangzhou Nutriera, and students and colleagues at the University of Guelph. Thank you very much.